Hello everyone. Today we are going to see the title of the unit is Microbes. Module name will be Economic Importance of Viruses and the module number is 8. So what is the basic outline? You will get to know what are viruses and their economic importance. So from here you will learn the definition of virus and what are their economic importance of viruses. So starting, what is a virus? So virus are very minute small particles which can infect a live host, that can be plants, animals, bacteria including human beings. So the structure of the virus will be, it will have a protein coat or an envelope which is called as a capsid, and in that it will have nucleic acid which will be either DNA or RNA. So now we will see the economic importance of viruses. So firstly we will see what are the positive aspects. Okay. First, the viruses will help in the eradication of crop pests. Okay. So viruses are very specific to specific insects. So this specificity of the viruses are used for their eradication through the crops. Okay, so the viruses can be naturally available on the plants, that is on the leaves, stem, roots, or they can be present in the soil. Okay, and they even can be commercially prepared, that is dilute solution of virus are made and this solution will be sprayed on the particular parts of the plant which is infected with the crop pest. Okay. So when the pest will ingest this virus, the virus will enter inside the body of the pest, they will reproduce inside and this will lead in the death of the pest. So second is the control blue-green algal bloom. So what is the algal bloom? It is nothing but the cyanobacteria which are very harmful because they produce some toxins, and which these toxins when are ingested by the humans or animals, this lead to the death. Okay. So viruses will attack the cyanobacteria and such viruses are called as cyanophages. These phages are very important to eradicate the bacteria which are present in the polluted water. So viruses help in cleaning of the water. Then third, we will see vaccine production. In the vaccine production, so viruses are used in the production of vaccines which control certain diseases like smallpox, polio, jaundice, etc. So there are three types of vaccines. Firstly, it is attenuated. In this, the viruses, live viruses are injected into the body of the host. So this will not form any diseases inside the body of the host. Example, chickenpox. Then secondly, inactivated or killed viruses. So here the viruses, killed viruses are directly injected inside the body of the host. Example is polio. And thirdly, it is subunits. Here the entire virus is not used. In fact, here the subunits or small units of the particular virus are used. Example is influenza. So what a vaccine will do, when a vaccine is given inside the body of the host, the antibodies will find it as a foreign antigen, okay, and they will produce antibodies which will destroy the viruses. So this will form a memory inside the host antibodies, okay, so this will help to prevent any further infection. Next, it is used in genetic engineering technology. So the viruses are used as genetic vectors. Okay, so what are vectors? Vectors are the carriers which will carry a particular gene of interest inside the body of the host cells. Okay, so virotherapy, it is called as a virotherapy. Here the genetically modified viruses are used to treat different diseases. For example, oncolytic viruses. These viruses are used to treat cancer cells. These viruses will particularly attack only the cancer cells and destroy them. Next, it's the bacteriophages. So what are bacteriophages? They are particularly the viruses which will infect the bacteria. Okay, so these bacteriophages are used in genetic engineering and genetic research. So if we see the mode of infection, particularly firstly the virus will find the host, that is the bacteria. It will get attached to it, then it will penetrate its DNA into the body of the host, where it will multiply. Then next is the DNA experiments. Here, two scientists, Hershey and Chase, they found that DNA was the genetic material, and they found it with the help of the bacteriophages. They did different experiments with the bacteriophages and found that DNA was the genetic material responsible. Then now we will see the negative aspects, how they are harmful. So the viruses will form various diseases inside the body of the host. Okay, they can form diseases inside the plants, animals and human beings. So this will damage various plant parts, that is leaves, roots, fruits, flowers, roots, etc. And this will lead to economic losses, because they will reduce the quality and quantity of the products. And loss on commercial scale. The phages will kill the beneficial microbes which are present in the antibiotics and milk products. So this will create a loss on the commercial scale. References. The books which are referred is Sangodkar and Chindarkar. Okay, so principles and practices for recombinant DNA technologies, and the following websites were used. Thank you.